Hello, and a very warm welcome to all of you. Whether you are a medical student, a clinical biochemist, a toxicologist, a forensic scientist, or simply a STEM enthusiast, I am absolutely thrilled to have you here. Today, we are diving into an explainer on something I find truly fascinating. We are looking at the application of enzymes in disease diagnosis. Specifically, we'll be exploring the biochemistry, the isoenzyme biology, and their real-world clinical significance. Let's dive right into this, shall we? We are going to map out a sort of molecular breadcrumb trail, one that actually helps us see the invisible.
Now, picture this clinical scenario. A patient rushes into the emergency department. They might be having an acute myocardial infarction, which is a heart attack, or perhaps they're suffering from severe toxic liver necrosis. From the outside, we only see the symptoms, but inside, right down at the cellular level, there is profound microscopic damage happening. The critical question for us is, how do we detect this invisible destruction accurately and, more importantly, quickly? We obviously can't just look inside the tissues with our naked eye. Well, for our clinicians and laboratory scientists, the answer is flowing right through the veins. We look at the blood, searching for highly specific biochemical signals.
To really understand the concept of non-functional plasma enzymes, I want you to think of our body's cells as highly specialized, localized factories. Each of these factories contains very specific machinery, and these are our intracellular enzymes. They do all their important work strictly inside the cell. However, when there is a sudden energy imbalance, maybe due to bacterial toxins, infective agents, or just a lack of oxygen, these factories sustain damage. Their structural walls, the cell membranes, become compromised. And what happens next? That internal machinery, those enzymes, leak right out through the broken membranes and wash into the bloodstream, which is essentially the river connecting all our organs. These are what we call non-functional plasma enzymes. You see, they have absolutely no job to do in the blood itself. They are only there because of normal cellular wear and tear, or in our case, pathological tissue damage. By measuring their sudden rise in the blood, we can figure out exactly which factory is failing.
And that brings us to our core cast of diagnostic enzymes. Think of this as our evidence map. Researchers have concentrated heavily on enzymes like creatine kinase, alanine transaminase, aspartate transaminase, acid phosphatase, alkaline phosphatase, and glutamyl transferase. Each one of these enzymes acts as a unique molecular breadcrumb. When we spot them in elevated numbers in the blood plasma, they point us directly back to the specific failing organs. So, whether we are dealing with jaundice, pancreatitis, cancer, or neurodegenerative disorders, these enzymes are our go-to markers. Let's start by taking a closer look at one of the body's most critical energy regulators.
Let's examine the biochemical reaction catalyzed by creatine kinase, or CK. For the biochemistry researchers in our audience, this is just a brilliant system. CK forms the absolute core of an energy network known as the phosphocreatine circuit. You can think of it as a cellular battery shuttle. During muscle contraction, our cellular energy, ATP, is consumed and turns into ADP. CK quickly steps in to rephosphorylate that ADP right back into ATP using phosphocreatine as the phosphate donor. It's a remarkably efficient, reversible shuttle system that perfectly maintains our cellular energy supply. But what happens when the muscle cells housing these little batteries are damaged?
When we compare the different CK isoenzymes, we see exactly why they are so incredibly useful for diagnostics. CK is a dimeric molecule, meaning it's made of M and B subunits, which combine to give us very distinct isoenzymes. This is where it gets highly specific. For example, CKMM predominates in our skeletal muscle. So, if this specific isoenzyme is elevated, we might be looking at muscular dystrophies or toxic myopathies. Then we have CKMB, which is heavily concentrated in the myocardium, the heart muscle. In patients suffering an acute myocardial infarction, the CKMB isoenzyme becomes a highly specific and accurate marker for heart damage. We also have CKBB, which is found mostly in the brain and spikes during brain injuries or a stroke. And finally, CKMT, which is located inside the mitochondria, closely coupled to the electron transport chain. By systematically mapping out which of these isoenzymes is elevated, a clinician can pinpoint the exact location of the tissue damage.
Now, here is a rather fascinating connection for those of you tracking infectious disease research. Did you know that high CK levels actually act as a prognostic marker for severe COVID-19? It's true. Because CK is a key marker of muscle damage, elevated levels are very frequently found in severe cases of COVID-19 as well as in deceased patients. What this tells us is that the viral infection is causing significant, widespread systemic damage.
This makes CK an incredibly important tool for doctors trying to gauge the clinical severity of the disease.
Moving right along, let's shift our focus to the liver sentinels. Alanine transaminase, or ALT, catalyzes the transfer of an amino group from alanine to alpha-ketoglutarate, which forms pyruvate and glutamate. For the toxicologists and clinical biochemists tuning in, you know this pathway is incredibly significant. While ALT is found in a few different tissues, it is present in its absolute highest concentrations right in the liver. Two forms exist, ALT1 and ALT2. When there is severe cellular necrosis in the liver, maybe from acute viral hepatitis B or C, or from toxic liver necrosis, this specific enzyme leaks out in large amounts. The rate of its activity in the serum gives us a very direct, reliable measure of liver function and injury.
So, a crucial point here is understanding the difference in specificity between ALT and another transaminase, AST, or aspartate transaminase. You see, while ALT is highly specific to the liver, AST has a much broader distribution across the body. AST is found in striated muscle, the heart, the liver, and the kidneys. Why does this distinction matter? Well, if we see elevated ALT without any other signs of liver disease, it might actually indicate muscle necrosis instead. Furthermore, for urological cancer researchers, monitoring both of these enzymes together is vital. Tumor cells in upper tract urothelial cancer, renal cell carcinoma, prostate cancer, and bladder cancer can all generate elevated levels of both ALT and AST in peripheral blood, which actually makes them quite useful in cancer prognosis.
And AST holds another surprising secret, particularly for the medical students and researchers studying rare diseases. AST serves as a biochemical diagnostic clue for infantile gangliocytosis. These are very rare, autosomal recessive, progressive neurodegenerative disorders like GM1 and GM2 gangliocytosis. They are caused by defects in the body's ability to degrade glycosphingolipids. Interestingly, in all of these patients, serum AST levels are notably elevated. It acts as a surprisingly simple but incredibly important biomarker for the initial diagnosis, follow-up, and monitoring of future therapies in these devastating neurodegenerative conditions.
Okay, let's examine two vital hepatobiliary markers next: alkaline phosphatase, or ALP, and gamma-glutamyl transferase, commonly known as GGT. For our clinicians, ALP is a very classic marker. It hydrolyzes phosphate esters at an alkaline pH. If a patient's bile ducts are blocked, which is a condition called cholestasis, ALP synthesis actually increases, and it gets solubilized into the blood by bile acids. But it is also critical for bone metabolism, meaning it rises during bone disorders like osteomalacia or even bone metastases. On the other hand, GGT is a truly fascinating marker for our pharmacologists and toxicologists. GGT metabolism is heavily centered on glutathione, which means it maintains our cellular defense against oxidative stress. Increased GGT is actually a major marker of antioxidant inadequacy. Elevated GGT is strongly associated with cardiovascular risk, hypertension, and even acts as an independent predictor of long-term mortality in heart attack patients. It tells us a very deep story about a patient's systemic oxidative stress.
Let's move to a marker that beautifully bridges the gap between clinical oncology and forensic science: acid phosphatase, or ACP. As the name suggests, ACP works in an acidic environment and has several isoforms. One specific isoform, prostatic acid phosphatase, or PAP, is strongly expressed by prostate cancer cells, particularly when the cancer metastasizes to the bone. But here is where it becomes incredibly relevant for the forensic scientists and law enforcement professionals joining us today. Because PAP is actively released and heavily concentrated in seminal fluid, measuring ACP is a critical, globally recognized tool used in forensic cases to identify seminal fluid. It is just a perfect example of how a single biochemical marker can span entirely different scientific disciplines.
Ultimately, measuring the minute changes in these leaked enzymes, our molecular breadcrumbs, offers a much greater degree of organ differentiation. We achieve a highly accurate, holistic map of the human body's pathological state. Unlike general inflammatory markers like albumin, these highly specific isoenzymes tell us exactly which factory is damaged, whether it is the liver, the heart, the bone, or the brain. They allow multidisciplinary scientists to diagnose, monitor, and treat their patients with incredible precision.
And so, I want to leave you all with a rather provocative thought. We have seen how enzymes like CK, ALT, AST, ALP, GGT, and ACP serve as absolutely critical markers across medicine, toxicology, and forensics. As our laboratory techniques become even more advanced and we map out more of these highly specific isoenzymes, how will these microscopic biocatalysts shape the very future of personalized medicine and real-time forensics?
The potential for earlier, far more precise diagnosis is honestly limitless. I strongly encourage you to think about how these powerful molecular breadcrumbs can be applied directly in your own specific fields of research and clinical practice. Thank you so much for joining me on this deep dive into diagnostic biochemistry. Keep exploring and keep asking those important questions.